Good afternoon, dear students. Today you have the first lecture in biochemistry. This lecture will be about biochemistry of proteins and basic principles of enzymology. These are our questions today. First of all, some words about what is biochemistry. I'll explain you in short way biochemistry of amino acids, protein structure, and peptides, the role of and functions of these molecules. But next, our three questions are the main. I'll explain today all about enzymes. What does mean enzymes? Characteristic of enzymes. Enzymes classification and structure, kinetics and mechanism of enzymes action, and how activity of enzymes can be regulated. First of all, what is biochemistry? Biochemistry studies the chemical process occurring in living organisms. Biochemistry is small chemistry, I want to say. And I want to say biochemistry is the daughter of physiology. Really, biochemistry has chemical basis, but you will learn chemical processes that are in our cells. We will discuss with you a structure of organic molecules that are in our organism. And first of all, we will learn with you processes that can be with these molecules. For example, we will study carbohydrates metabolism, and we will discuss how glucose is transformed into lactate. And I hope you know this feeling when you did physical exercises. After it, you can feel not well because glucose in anaerobic conditions will be transformed into lactate. And this process we will study with you, we will discuss. And not only this, biochemistry studies a lot of different processes, and for every of this process we will learn enzymes, because without enzymes processes cannot exist in our organism. We will start from biochemistry of proteins, and first of all I'll say some words about biochemistry of amino acids, because amino acids are structural blocks for proteins and peptides. And amino acids are joined together by peptide bonds in the structure of proteins and peptides. What does mean proteins and what does mean peptides? Peptides are polymers with less than 100 amino acids. If more than 100 amino acids, we can say about protein. You can find here the common structure for amino acids. In alpha position, every amino acid has carboxylic group, and that is why this compound is acid and amino group. You should know the structures of 20 standard amino acids that are involved in building of proteins. And practically, if you know this part of amino acids, a common part of amino acid, you should study only side radical of amino acids. You must know chemical structures of 20 standard amino acids. Again, this is a common structure, carboxylic group and amino group, and we will discuss today that carboxylic group gives acidic properties and amino group gives basic properties for amino acids. In our cells, there are proteinogenic amino acids. That amino acids, as I said, are used for building of proteins and peptides, and non-proteinogenic amino acids. Proteinogenic amino acids are present in our cells in free form and can be used for synthesis different biologically active compounds. I hope you know, for example, tyrosine. Tyrosine is standard amino acid that can be involved in protein synthesis, but in the same time, tyrosine is used for the synthesis of hormones like epinephrine, norepinephrine, T3, and T4. Non-proteinogenic amino acids are involved in metabolism, and some of them can be as intermediates of metabolic processes. There is biological classification of amino acids. For biochemistry, this classification is important. According to this classification, there are two groups: essential amino acids and non-essential amino acids. Essential amino acids are not synthesized in our organism, and these amino acids must be present in our diet. These amino acids are very important not only for building of our proteins. Some of them are used in another metabolic processes. For example, methionine. Methionine is essential amino acid that necessary for synthesis of phospholipids of our membrane, especially phosphatidylcholine, or more old name is lecithin. Without methionine, this phospholipid, the main phospholipid of our membrane, cannot be synthesized. If in organism there is low quantity of methionine, steatosis can occur. Steatosis is fatty liver, because if methionine is in deficiency, membranes cannot be fixed normally. Non-essential amino acids are synthesized in our organism, and practically with these amino acids, we don't have any problems, because different intermediates can be transformed into these amino acids. For biochemistry, another classification is very important. This classification is based on polarity and charge of side radical. You must know that there are 4 classes of amino acids. First class, non-polar amino acids. In the structure of these amino acids, side radical doesn't have polar or charged groups. Polar amino acids without any charge, non-charged amino acids. Inside radical of these amino acids, you can find polar group like OH, SH group, but you cannot find charge. Polar amino acids with positive charge or positive charged amino acids. These amino acids have alkaline properties, because in side radical you can find additional amino group. Polar amino acids with negative charged or negative charged amino acids, these amino acids have additional carboxylic group in side radical. Here you can find the illustration of structure and quantity of amino acids for every class: non-polar amino acids, polar non-charged amino acids, negative charged amino acids, and positive charged amino acids. You should know not only radical, this is only radicals, side radicals of amino acid. You should know all structure of every amino acid. You can find here the information about general properties of amino acids. These properties, I hope, are very good from biochemical chemistry. For biochemistry, next properties are more important. First property that you must know: acid-base properties of amino acids, or amphoteric properties. Amino acids, every amino acid in alpha position has carboxylic group and amino group. Carboxylic group can work as the acid, can give in environment protons. Amino group can work as a basic group, and that is why amino acids are amphoteric in nature. Amino acids can work as an acid, as a base, and can be ionized in different conditions. You can find here that every amino acid can form a sweeter ion, but unfortunately I see mistake here. NH3 plus should be here. Sweeter ion is non-charged ion, because quantity of pluses is equal to quantity of minuses. For every amino acid, there is such point as the isoelectric point, or pi. What does mean isoelectric point? Isoelectric point or pi is the pH when all, which is very important, all molecules of amino acid exist in form of zeta ion. All molecules, practically all, 99% of all amino acids don't have any charge. Charge is equal to 0. For every amino acid, we can find information about pi. But generally, we can say for hydrophobic amino acids or amino acids of first class, pi, middle pi, approximately is equal to 6. For non-polar, polar non-charged amino acid, pi is approximately 5.5. For acidic amino acid, pi is lower than 7, approximately 3. For basic amino acids, positive charged amino acids, pi is approximately 9. For histidine, pi is equal 7. And you can orientate in middle numbers for pi in every class, because we will solve, we will do special tasks with pi on practical classes. Next property, amino acids can form peptide bonds. Peptide bond is formed between carboxylic group in alpha position of first amino acid and amino group in alpha position of the next amino acid. For correct structure of dipeptide, tripeptide, we should write the structure from amino group. Amino group constantly will be the first and will be free, because in a living organism, the first amino acid that involved in protein synthesis has free amino group, and carboxylic group participates in formation of first peptide bond. And releasing of water can give a covalent bond, but this covalent bond is peptide bond and has specific properties. For example, this bond is shorter than standard covalent bond. About properties of peptide bond, you can read more in your textbooks. We are limited in time. Next property, stereochemical properties. Amino acids in cells can be in nature, can be in L and D form, in form of L, stereoisomers, and D. How you can write L amino acid and D amino acid correctly? If you will write carboxylic group in the top and radical here, and if you will write amino group on the left, this is L amino acid. If this amino acid look into a mirror and can find that amino group will be in another position. On the right, this is D amino acid. Next, our question, protein structure and properties. Protein molecules are the main molecules in living organism, and protein structure we can present in 4 levels. So there are 4 levels of protein structure: primary, secondary, tertiary, and quaternary structure. The illustration of these structures you can find here. This is primary, secondary, tertiary, and quaternary structure. Primary is the sequence of amino acids. These rounds are amino acids that connected is bonded by peptide bonds. Secondary structure, this polypeptide chain can form alpha helix or beta structure. Tertiary structure is a 3-dimensional structure, and polypeptide chain after formation alpha or beta structure can form globular or fibrillar. Some proteins can be only with 3 levels of structural organization, but we can find a lot of proteins with next level of structural organization. This is quaternary structure. Some polypeptide chains can be connected by non-covalent covalent bonds. These proteins are oligomeric proteins. If protein has 2 or more polypeptide chain, 2 or more subunits, we can say about oligomeric structure. For example, if myoglobin has only 3 levels of structural organization, hemoglobin has 4 subunits, has 4 polypeptide chain, and we can say about quaternary structure for hemoglobin. Stabilization of these structures can be as a by covalent bond, like primary structure, as a non-covalent bond. We will discuss this question on the practical class. Next, not only proteins are built by amino acids, peptides. If a quantity of amino acids in polypeptide chain less than 100 amino acids, we can say about peptides. For peptides classification, you should know the classification of peptides according to the functions of these molecules. For example, in our organism, there are hormones, oxytocin and vasopressin, neuropeptides, for example, opioid peptides. Alkaloids can be used for treatment of your future patients, antibiotics, toxins, regulatory peptides. Not all functions are here, but main functions of peptides you must know. Next, very important, long, big question, but I'll try to explain it in more short form. Properties of proteins. First of all, proteins have high molecular weight, and that is why proteins form colloidal solutions. Proteins are amphoteric in nature. They can work as an acid, as a base, and that is why for every good known protein we can find in any book the number of isoelectric point, pi. I want to say again, pi is pH when all molecules of protein don't have any charge. Charge is equal 0. Proteins act as a buffer solutions. My example is protein buffer system. This system in blood is necessary for maintenance of pH. Denaturation, this is destruction of secondary, tertiary, and quaternary structure of proteins. Primary structure will be existed. If primary structure will be destroyed, we can say about hydrolysis. Precipitation by neutral salts or salting out. Next property is very important for medicine because this property can be used in special method. This method is called electrophoresis, electrophoretic movement. Proteins can move in constant electric field, and the velocity of movement depends on molecular weight and charge. If protein has low molecular weight and high charge, the velocity of movement will be very high. This property is used in such method as a serum protein electrophoresis. This method is used as additional method for diagnostic. You should remember this picture, and we will discuss this picture, especially in the next semester when we will study with you biochemistry of blood. But now you should remember the shape of electrophoresis, and you must remember that in serum of our blood we can find by electrophoresis on the paper 5 fractions. First of all, albumins and globulins, globulins alpha 1, globulins alpha 2, beta, and gamma. The size of peak depends on quantity of this fraction in serum of blood. The common concentration of common protein in blood is equal 65 to 85 grams per liter. Approximately 50% of all proteins in serum blood, these are albumins, and which is why we can find such big peak of albumins. Next our part will be part of our lecture will be about enzymology. I said we will learn with you reactions that can be in organism For every reaction in our organism, we can find enzyme. What does mean enzyme? Enzymes are biocatalysts. What does mean biocatalysis? Enzymes are present only in living cells, and that is why biocatalysis enzymes increase the velocity of reaction that catalyzed by these enzymes. And next, enzymes are biocatalysis that protein in nature. There is such rule. Practically all, and we will think, all proteins in our organism, all enzymes in our organism are proteins. In next semester, we will discuss with you functions of RNA and DNA, and we can say some RNA can be with catalytic functions. But now and in future, we will use the rule enzymes are proteins in nature. Enzymes are biocatalysis that proteins in nature. You should know such terms in enzymology. First of all, here we can find these terms. Compound that will be transformed by enzyme is called substrate. A substrate is transformed by enzyme into another compound. This another compound is called product. Substrate, enzyme, and product. Here you can find that every enzyme has active site. This active site is specific place where substrate is transformed into product. This active site is formed by amino acids and functional groups of amino acids participate in this transformation. About nomenclature enzyme, how we can know this name is name of enzyme or not? If you will find suffix "ase", you should be sure this is the name of enzyme. For example, you know that milk sugar as lactose. Lactose is split in our intestine by lactase. Maltose is split by maltase. But we can find a lot of names of enzymes without any ase because these enzymes were named maybe before the formation of this simple nomenclature. For example, pepsin or trypsin, we know they are enzymes, but without any name. The International Union of Biochemistry initiated standards of enzyme nomenclature, which recommended that enzyme names indicate both as a substrate name as a type of reaction. And that is why in enzyme name you can find the name of substrate and the type of reaction. Here you can find example. Lactate is transformed into pyruvate. This is reversible reaction. And enzyme is called lactate. Lactate is substrate. Dehydrogenase. There is dehydrogenation of lactate. Lactate will lose protons and will be transformed into pyruvate. And we can find suffix "ase". Next, classification of enzymes. The enzymes are classified according to the type of reactions that which enzyme catalyzes. I want to say there are 6 classes of enzymes. You can find here classes of enzymes. The list of classes cannot be changed. Oxidoreductases, this is the first class constantly. Transferases, this is the second class. Hydrolases, class number 3. Isomerases, class number 4. Lyases, class number 5. And ligases or synthases, this is last class number 6. Here you can find which reactions are catalyzed by enzymes of every class. And you should know examples of enzymes for every class. For example, oxidoreductases. Oxidoreductases catalyze the reaction of oxidation and reduction. Example is here alcohol dehydrogenase. Ethanol is transformed into acetaldehyde. But this is only one reaction. In our organism, there are different subclasses of oxidoreductases. By the way, every enzyme has a specific code. In this code, we can find 4 numbers. First number is number of class, second number is number of subclass, third number is number of subclass, and last fourth number is number of enzyme in subclass. And here you can find different subclasses of oxidoreductases, but this is only example. When we will discuss with you bioenergetic, we will discuss about these subclasses more. Next, this is example of transferase, alanine aminotransferase. By the way, this reaction is very important in metabolism of amino acid, and activity of this enzyme is used for diagnostic. Alanine aminotransferase is specific for hepatocytes. In the case of problems with hepatocytes, alanine aminotransferase activity in blood will be increased. What about the structure of enzymes? Enzymes are proteins in nature, and I hope from biochemical chemistry you remember proteins can be as simple as a complex protein. Simple proteins have only protein part in the structure. Complex proteins have as a protein as a non-protein part, and according to this non-protein part, complex proteins can be divided on six classes. For example, chromoproteins, lipoproteins, glycoproteins, and other. There are six classes of simple proteins, and there are six classes of complex proteins. You know now that there are six classes of enzymes. And these enzymes of these classes can be as simple as a complex. What does mean complex enzyme? Here you can find the illustration for this. A complex enzyme has protein part. This protein part is called upper enzyme, and non-protein part is called cofactor, and complex enzyme is called Hola enzyme. I said there are six classes of enzymes, where in which class of enzyme we can find simple enzymes without any cofactor, only protein part these enzymes have. You should remember all classes, six classes of enzymes practically are complex enzymes. These enzymes can work if there is non-protein part. Only hydrolases, in hydrolases we can name pepsin, chymotrypsin. Enzymes of this class of hydrolases, are simple enzymes. In our organism, we can find more quantity of complex enzymes than simple. And now here you can find the structure of complex enzyme. And I want to say about cofactor. Cofactor can be as an inorganic molecule as an organic molecule. If this molecule is inorganic molecule, like a metal, ion of metal, this non-protein part can be called as a cofactor. But if cofactor is small organic molecule, this small organic molecule is non-protein, absolutely. If cofactor is small organic molecule that binded with protein part covalently and cannot be separated from protein part, this cofactor can be called as a prosthetic group. Again, prosthetic group is organic part of complex enzyme that binded with peptide part of enzyme covalently and cannot be separated from protein part. Coenzymes, as a rule, we will say about coenzymes. Coenzymes are organic molecules too, small organic molecules too, but these molecules are connected with protein part non-covalently and can be separated from protein part. I want to say, as a rule, coenzymes are vitamins. You know, water-soluble vitamins are very useful for our organism, but our organism cannot see vitamin B1, B2, B3, B9 without transformation of these vitamins into coenzymes. Examples of coenzymes you can find here: FAD, FMN, NAD, NADP, FAD and FMN are derivatives of B2. B2 is transformed in our organism, vitamin B2 in FAD or FMN. Vitamin PP or niacin is transformed into NAD and NADP. These are coenzymes. For example, here you can find ethanol is transformed into acetaldehyde with participation of NAD. NAD is coenzyme of alcohol dehydrogenase. The structure of the enzyme. Here you can find, in every enzyme, we can find specific site. This site is called active site. This is the illustration how in active site, substrate is transformed into product. Substrate and product. Substrate and product. Every enzyme has active site. In this site, we can find a catalytic part for binding with substrate. If this enzyme is complex, in active site, we can find coenzyme, and without coenzyme, substrate cannot be transformed into product. But some enzymes can be with another site. And this site is located on another subunits. As a rule, oligomeric enzymes, you know what does mean oligomeric proteins. Oligomeric enzymes can be with another part. This site is called allosteric site or regulatory site. Here you can find enzyme that has two subunits. One subunit has active site, and this subunit is called catalytic subunit. Another subunit has allosteric site, and this is regulatory subunit. This active site, substrate should be connected with regulatory subunit. Specific molecules, modificators, can be connected. These modificators can work as inhibitors or activators. Inhibitors decrease the activity of enzyme because there is changing of active site. Activators stimulate the work of active site, and enzyme will work better. Next question. Our velocity is very fast, like velocity of enzymes reaction, because we don't have enough time for all questions. And next, my question, some words about kinetics and mechanism of enzymes action. You can find here photos of two scientists, Michaelis and Menton. These two scientists studied kinetic of enzymatic reaction. Kinetic enzyme kinetic studies the chemical reactions that catalyzed by enzymes. And this part of biochemistry studies how velocity of chemical reactions in our organism depend on different factors. In enzymology, we should understand velocity of reaction can give information about activity of enzymes. For analyzing, if you want to do a conclusion according activity of enzymes, we should do the analysis of velocity of chemical reaction. And if you will find that velocity of chemical reaction is higher, we can say activity of enzyme is higher. And that is why if I'll say activity of enzyme, you should understand I can say velocity of reaction, because these are similar practically in understanding. If velocity of reaction is high, we can say about high activity. We cannot say in enzymology about quantity of enzymes. It is not interesting because, as a rule, some molecules of enzymes can be active, some molecules can be inactivated. And we want to know about active molecules of enzymes. But first of all, you should remember activity of enzyme. In this case, we can say about velocity of reaction. Velocity of reaction or activity of enzyme depends on different factors. First of all, concentration of substrate. You should know how you can draw this graph, and there is special Michaelis-Menten kinetic equation. In your textbook, you can find the creation of this equation, but you know this equation without additional reactions. This equation is illustrated by this graph. Velocity of reaction depends on maximal velocity, concentration of substrate, and in this equation you can find Michaelis constant. Michaelis constant plus concentration of substrate, and that is why logically Michaelis constant, this is concentration of substrate, and we can find here point of Km, Michaelis constant. Michaelis constant is the concentration of substrate when velocity of reaction is equal half of maximal velocity. How we can find Km for every enzyme? We should find half of maximal velocity, and after it, in this graph, we can find without any problems Michaelis constant. This graph has such shape because in environment, as a rule, there is limited quantity of active sites, limited quantity of molecules of enzymes. And here, if you will add substrate, velocity will be increased because we have free active sites of enzymes. And after it, velocity will be increased before maximal velocity. And here you can find moment when velocity will be maximal and cannot be changed because all molecules of enzymes, all active sites are busy by substrates. We can say that enzymes molecules are saturated by substrates molecules, and free active sites are absent. Velocity of reaction or activity of enzyme depends on pH. In our organism, as a rule, enzymes work in neutral pH. We can find there is set point as the optimal pH. Optimal pH is pH when velocity of reaction is maximal or activity of enzyme is maximal. But some enzymes can be this optimal pH in acidic environment or in alkaline environment. My example is, for example, pepsin has optimal pH from 1.5 to 2, or trypsin that very active in alkaline conditions. Why velocity of reaction or activity of enzyme depends on pH? The correct answer is because pH can change the rate of ionization of functional groups in active site of enzyme. pH can change the rate of ionization of functional groups in active site of enzyme. In active site, I said there are functional groups of amino acids, carboxylic group, amino group, and pH can change the ionization of this group. And that is why enzyme can be with different activity in different pH. By the way, pH can change the rate of ionization of functional group in molecule of substrates, and this ionization can change the velocity of reaction too. Next, temperature. Temperature is the next factor that can change the velocity of reaction. Velocity of reaction or activity of enzyme depend on temperature. As for pH, we can find optimal temperature for our enzymes, for enzymes of our body. As a rule, optimal pH is here, 36.60 degree. If temperature will be increased or decreased, conformation changing can change the velocity of reaction. We cannot say about denaturation because denaturation can be maybe here, can start here, but we know we cannot feel good if concentration is not optimal for our body. If concentration is increased and temperature, if temperature is increased, and temperature will be more than 38 degree, we cannot feel well. And we should use special pills, because if temperature of body will be increased, there is the conformation changing of our enzymes, and these enzymes cannot work optimally, because this is the temperature. This is a temperature that not optimal for the work of our enzymes. Next, some words about mechanisms of enzymes action. Substrate, we know, is transformed into product, but for this transformation, substrate should react with enzyme. And first of all, enzyme-substrate complex should be formed. After it, this complex can be activated, or maybe some active forms can be formed, because there is changing of substrate in active site. And after it, there is formation of product inside of active site. And after it, product will be separated from enzyme. Enzyme cannot be changed in this case, only substrate can be changed, and enzyme can transform the next molecule of substrate into product. If you want to explain mechanism, we should start from explanation. When enzyme react with substrate, there is formation of enzyme-substrate complex. This is key moment in enzymology. After this substrate will be transformed into product, enzyme will catalyze the next transformation. How we can explain why enzymes can increase the velocity of reaction? Enzymes, thermodynamically, how we can explain it? Enzymes can increase the velocity of reaction because enzymes reduce the activation energy. Here you can find it. Enzymes reduce the activation energy. What does mean activation energy? Activation energy is additional energy that necessary for transformation of substrate molecule into active state. From this active state, molecules of substrate can be transformed into product. And now I can explain it by this graph. As a reactant, this is substrate. As a product, it has specific level of energy. As a rule, level of energy of substrate is more than level of energy of products. Here you can find two graphs. First graph illustrates how substrate is transformed into product without enzyme. And where we can find here activation energy. Activation energy, as I said, this is additional energy that can be used for transformation of molecules of substrate into active state. And that is why this energy is activation energy. You can find it here, activation energy. If in the environment there is enzyme, activation energy will be reduced. And more quantity of molecules of substrates can be activated and can be transformed into product. If more quantity of molecules of substrate will be activated and can be transformed into product, we can find increasing velocity of reaction. Without enzymes, not all molecules of substrate can be activated. Not all molecules of substrate can take this additional quantity of energy, and that is why it cannot be transformed into product. And that is why enzymes increases the velocity of reactions, because enzymes decrease the activation energy. Next, there are different explanations why enzyme can transform substrate into product. And there are some models that can explain this mechanism. First, the simplest model, but this is only hypothesis, lock and key hypothesis. And according to this hypothesis, substrate can work as a key for the lock. Lock is enzyme. Practically, the shape of substrate is similar to the shape of enzyme. But this hypothesis can explain that specificity of action for enzyme as absolute specificity. According explanation, what does mean specificity? In our organism, for every substrate should be one enzyme, but there is relative specificity because some enzymes can transform some substrates. One enzyme can transform some substrates. For relative specificity, this hypothesis is not good because in this case we can find one enzyme and one substrate can react each other. But for example, if you will take pepsin, pepsin can hydrolyze not only one kind of proteins. There are different kinds of proteins, or lipase can act on different kinds of triacylglycerols. And it is why lock and key model, this is only hypothesis. Induced fit model is more good understandable and can be used for explanation how enzyme can catalyze different transformation of different substrates. According to this theory, in this case I want to say about theory, if a substrate reacts with active site, there is changing as a shape of substrate, as a shape of active site, and that is why one enzyme can transform different substrates. As I said, for pepsin or lipase or amylase, for example. You should know how we can explain enzymatic catalysis on molecular level. We said general explanations, but on molecular level, there are some effects, and these effects can be realized in different enzymes. You should know there are 4 effects, molecular effects of enzymatic action. Orientation effect, strain effect, acid-base catalysis, and covalent catalysis. We will discuss it more on practical classes. Not all these effects can be realized for one enzyme. One enzyme can catalyze transformation of substrate into product because, for example, 2 or 3 these molecular effects can be realized. And next, our question, this is regulation of enzymatic reactions. In our organism, all processes are regulated. And first of all, I want to say about inhibitors and activators. Inhibitors reduce the activity of enzyme. Activators increase the activity of enzymes. First of all, about inhibition. Inhibitors are specific molecules that can reduce the velocity of reactions in our organism. There are different types of inhibitors, and it is why there are different types of inhibition. First of all, you should know there is irreversible inhibitions. And there are irreversible inhibitors. These molecules of irreversible inhibitors bind to enzyme through covalent bonds, and these molecules are poisons. After such inhibition, the enzyme cannot be reactivated. This enzyme, after binding with such type of inhibitor, cannot work. But there are reversible inhibitors. Reversible inhibitors are connected with enzymes non-covalently, and that is why this process can be reversed as a reversible process. The enzyme after binding with reversible inhibitor can be reactivated. Reversible inhibitors can be divided to two groups. Reversible inhibition can be competitive and non-competitive. Here you can find the example of irreversible inhibitor. For example, diisopropyl phosphate is poison. This is toxic compound, and you can find that in active site of acetylcholinesterase, this inhibitor forms covalent bond between OH group of serine in active site of this molecule, and acetylcholinesterase can be inhibited. But some poisons of acetylcholinesterase can be used in treatment of some diseases. For example, physostigmine is an irreversible inhibitor, but physostigmine can be used for treatment such as inherited disease as a myasthenia gravis. We will talk about it more on practical classes. I said reversible inhibition can be as a competitive as a non-competitive. If we will say about competitive inhibitor has a similar structure to substrate. You can find here act enzyme with active site, and shape of these two molecules are similar. And this inhibition is reversible. Why? Because inhibition depends on quantity of substrate. If concentration of substrate will be increased, competitive inhibitor will be removed from active site. As a rule, if there is such type of inhibition, we can reactivate enzyme very fast by adding molecules of substrate. This principle is used for treatment of methanol poisoning. Methanol is toxic compound. Ethanol can be metabolized in our organism by alcohol dehydrogenase. Methanol cannot be metabolized in our organism and must be inhibited. Alcohol dehydrogenase is an enzyme for metabolism of ethanol. If alcohol dehydrogenase will be inhibited by methanol, we can reactivate alcohol dehydrogenase by higher quantity of ethanol. Practically, methanol poisoning we can treat by toxic doses of ethanol, but we should do it in time. Next, non-competitive inhibition. In this case, this shape of inhibitor is not like shape of substrate, and non-competitive inhibitor can be connected with enzyme, and enzyme will be reactivated. And in competitive inhibition can react with enzyme in another place, non-inactive site. In this case, you can find active site will be closed. How we can reactivate enzyme in this case? In this case, we should use specific molecules. These molecules are reactivators. As a rule, these molecules have S8 groups. These are S8 complexons, and practically these S8 groups can take inhibitor and can remove from enzyme, and this enzyme will be reactivated. Next, I want to say about feedback inhibition. Feedback inhibition is inhibition by final product. You can find here illustration. As a rule, in our organism, in process, we can find some reactions, and it is why there are some enzymes. You know, in our organism, for example, glucose can be transformed into lactate. Glucose is a start product, is this substrate of process, and lactate is product. But between glucose and lactate, there are 11 reactions, and that is why there are 11 enzymes. Some of these enzymes work as a key enzymes. These enzymes are regulatory enzymes. These enzymes can be activated or inhibited. And in our metabolic processes, this is not for glucose metabolism, different processes. If product is not used, especially if this is a synthesis of compound, if product is not used, this product will work as an inhibitor for enzymes. Two first enzymes, as a rule, are key enzymes, regulatory enzymes, and these enzymes or enzyme can be blocked by product. And this inhibition is called feedback inhibition, the inhibition by end final product. You know that in our organism, for regulation of enzymatic processes, there are two pathways, and every of these pathways has own mechanisms. And that is why you should understand there are pathways for enzyme regulation, and every pathway has mechanisms. So there are two main pathways. First of all, regulation of amount of enzyme. How we can change the velocity of all process, as you saw, for example, one compound is transformed into another, and another. We can change the quantity of enzyme. If quantity of enzyme, if synthesis of this enzyme will be stimulated, you should be sure these processes will be very, very active. Another pathway is the regulation of enzyme activity. In this case, we can say that quantity of enzyme cannot be changed, amount of enzyme cannot be changed, but we can activate or we can inactivate enzymes or enzyme. We can stimulate or inhibit the work of enzyme. And that is why first pathway, this pathway is about amount of enzyme, another pathway is about enzymes activity. And now mechanism of first pathway, how we can change amount of enzyme. We can stimulate or induce the synthesis of enzyme, or we can repress the synthesis of enzyme. And that is why there is induction and repression. If enzyme is necessary, if, for example, substrate will go into cells, synthesis of enzyme will be activated. For example, ethanol can stimulate the synthesis of alcohol dehydrogenase, or phenobarbital can stimulate the synthesis of hydrolase like cytochrome P450, and this mechanism is good understandable in our organism. And repression, if enzyme is not necessary, if enzyme cannot synthesize this molecule in this time, there is a repression, inhibition of synthesis of this enzyme on genes level. Next, how enzymatic activity can be regulated. You can find here mechanisms: allosteric regulation, reversible covalent modification, part proteolysis, proteolytic activity. Enzyme's activity can be regulated by another proteins, but I want to say first of all about these mechanisms. What does mean allosteric regulation? I said in our organism there are specific allosteric enzymes. These enzymes have not only active site, these enzymes have allosteric site, regulated site. With this site, inhibitors or activators can be connected, and if allosteric site will be binded, for example, with inhibitor, in this case, active site cannot work normally and velocity of reaction will be decreased. Allosteric enzymes, as a rule, they are oligomeric enzymes. Kinetic of allosteric enzymes is different because for allosteric enzymes we can find cooperative effect, and this effect we'll discuss in practical classes. Next, what does mean reversible covalent modification? Activity of some enzymes can be changed by adding of functional group, and this functional group is connected covalently. We can say now about phosphorylation and dephosphorylation of enzymes. Two molecules of enzyme phosphate group can be added or can be removed, and activity of enzyme can be changed. This is not inactivation or activation for different enzyme. Phosphorylation or dephosphorylation can lead to different effect. And next mechanism, proteolytic activation. This mechanism is good known for you. I hope you know. pepsin, for example, is synthesized in inactive form, in form of pepsinodine. If part of pepsinodine molecule will be removed hydrolytically by hydrolysis, pepsinodine is transformed into active pepsin. And again, I want to say we will discuss it more in practical classes. And last, my question, I want to say some words about isoenzymes. Isoenzymes are multiple forms of one enzyme. In our enzyme, in organism, for example, there is lactate dehydrogenase. Lactate dehydrogenase catalyzes transformation of lactate into pyruvate. But for this enzyme, in different tissues, we can find different forms. But these forms catalyze one reaction: lactate pyruvate, pyruvate lactate. And it is why isoenzymes are multiple forms of one enzyme. These forms are differ in electrophoretic mobility, kinetic properties, structure, and that is why kinetic property structure is different, and that is why kinetic properties are different. Some isoenzyme can catalyze, can move reaction in one side or another side. Lactate dehydrogenase 1, for example, catalyzes transformation of lactate into pyruvates more than pyruvate into lactate. And it is why as enzymes are multiple forms of one enzyme that differ in structure, physical, chemical, and catalytic properties. You should know these examples. Lactate dehydrogenase. Lactate dehydrogenase in our organism can be in 5 multi-enzyme forms. There are 5 isoenzymes: LDH1, 2, 3, 4, and 5. Lactate dehydrogenase in the structure, this is oligomeric enzyme, has 4 subunits of 2 types. In the structure of lactate dehydrogenase, H subunits and M subunits can be present, but some quantity is formed. Lactate dehydrogenase 1 has only 8 subunits. And this isoenzyme you can find, first of all, in heart. LDH5 has only M subunits. This is the main enzyme form for skeletal muscles. And another subunit, this is the combination of H and M subunits. LDH2, H3M, here I see mistake, LDH3, 2H and 2M, LDH4, 1H and 3M subunits. And if there is problem with organ where this isoenzyme is located or has maximal activity, in this case in blood we can find increased activity of this enzyme. For example, LDH1 and 2, this is enzyme for heart. In the case of myocardial infarction, we can find increased activity in blood of these isoenzymes. LDH4 and 5, these are muscles, isoenzyme or liver, and that is why definition of activity of these isoenzymes form we can find for diagnostic of different organs. Not only lactate dehydrogenase can be a good example of isoenzyme. In our organism, there is creatine phosphokinase or creatine kinase. This enzyme has 3 isoenzyme forms. This enzyme is oligomeric enzyme too, but this enzyme has only 2 subunits, M or B. And combination of these subunits can give the different isoenzymes. There are 2 kinds of subunits, M and B. M is muscles, B is brain. Combination M and M is located in skeletal muscles. MB is in heart, BB is in brain. And definition of activity of creatine phosphokinase, total activity of creatine phosphokinase in blood, and definition of activity of isoenzymes can be used for diagnostic of different diseases, especially if the activity of MB CPK will be increased more than 5% from total activity of enzyme, we can say that maybe this patient has myocardial infarction. Thank you, dear students, very much. This is my photo. I like this photo. And this is your first lecture. And as a rule, I can imagine students on the first lecture like these small birds, but you should be sure if you will be hard-working students, you can transform in beautiful birds and you can fly really. Thank you very much.